Hello everyone, welcome to the ITLS Academy. Today I'll discuss about the pH meter, the fifth instrument under the bioinstrumentation topic. In the previous slide, we have learned about the autoclave sterilization technique, hot air oven, the incubator, the laminar airflow, and now today I'll be taking the topic pH meter. Okay, before moving towards the instrument, the device pH meter, we have to understand what does the pH means. What is the pH of a solution refers to. Let's begin with pH is defined as minus log H ion concentration, where H ion is the molar hydrogen ion concentration. Actually, pH must be defined in terms of thermodynamic activity of H ion and not as the molar concentration. Activity depends on the concentration of other electrolytes present in the solution. However, in biological systems, the total ionic strength is always sufficiently low that the effect of electrolyte concentration on pH is small enough to be ignored. It is a general rule of biochemistry that the activity for all the practical purposes equals the concentration. Whereas this is not always the case, the rule is almost universally applied. The pH was originally an acronym for the French word, the French clause, that is the power hydrogen, which can be translated into the English as the power of hydrogen, or we can say that the potential of hydrogen. Hence, the name has occurred, and we call it in the short form as the pH. The pH is used to quantify the acidic or alkaline nature of a water soluble substance, which is measured in terms of H ion concentration. Actually, pH the discovery of pH was based on the taking the normal solution, the water, as a solution for measuring the pH of water, and the H ion concentration describes the power of hydrogen. The pH value is an expression of the ratio of H ion to hydroxide ion. Hence, if the H ion is greater than the hydroxide ion, the solution is called to be the acidic. Otherwise, if the OH ion concentration is more in solution as compared to the H ion concentration, the solution is known as the basic solution. A pH value is a number from 1 to 14 with 7 in between as the middle point, which is denoted as the neutral point of the pH. Values below 7 indicates the acidity, which increases as the number decreases, and and the value of 1 being the most acidic in nature. And we can say that the values above 7 indicates the basicity of any solution, which increases as the number increases from 14, as the number increases from 7 to 14, and the last one, the 14, is the most basic in nature of that particular solution or any substance. Okay, let's now understand the see the introduction of pH meter. A pH meter is an electric device used to measure hydrogen ion activity, acidity or alkalinity in water based solution, indicating its acidity or alkalinity expressed as the pH value. A pH meter is an electronic instrument used for measuring the pH of a liquid. Liquid can be of any material. It does not mean that always we have to check the pH of the water. A typical pH meter consists of a special measuring probe, that is a glass probe, and a reference electrode, and which are connected to an electronic meter that measures and displays the pH reading while calibrating the pH. It provides a value as to how acidic or alkaline a liquid is. For example, acid dissolves in water and forms a positively charged hydrogen ion. The greater this concentration of hydrogen ion, the stronger will be the acid, or the stronger will be the acidic solution. Fundamentally, a pH meter consists of a potentiometer attached to a pH responsive electrode and a reference unvarying electrode. The difference in this electrical potential relates the acidity or pH of the solution. Okay, let's see the working principle of the instrument. A pH meter measures the voltage between the two electrodes placed in the solution. The potential of one electrode, that is the pH electrode, is pH dependent. The action of this electrode is based upon the fact that certain types of borosilicate glass are permeable to H ion concentration but not to the other cation and anion. Therefore, a thin layer of we can say 0.05 to 0.1 mm of such glass is interposed between the two solutions of different H ion concentration. This H ion will move across the glass from the solution of high to that of low H ion concentration. Since the passage of H ion through the glass adds a positive ion in the solution of low H ion concentration and leaves behind a negative ion, an electric potential develops across the glass as the H ion concentration moves from higher to lower position. When a pair of electrode or combined electrode is dipped in an aqueous solution, a potential is developed across a thin glass of the bulb. The EMF of complete cell formed by linking of these two electrodes at a given solution temperature is therefore EMF is equals to electrode reference and electrode of glass. Here E reference refers to the potential of a stable calomel electrode which at normal room temperature is 0.5250 volt, and E glass the potential of glass electrode which depends on the pH of the solution under test. A pH meter acts as a voltmeter that measures the electrical potential difference between the pH electrode and the reference electrode and displays this result in terms of pH value of the solution in which they were immersed. That in whatever we are taking the solution, the difference between the potential electrical potential of the electrode and reference electrode is measured in terms of the pH. An electrical potential develops when one liquid is brought into contact with the another one, but a membrane is needed to keep such liquids apart. Okay, what are the materials and methods for the sample analysis? A pH measurement system consists of two probes, a buffer powder or capsule for calibration, and third is the high input meter. We need a high input meter, and we also need a distilled water. The pH measuring electrode is a hydrogen ion sensitive glass bulb. The reference electrode output does not vary with the activity of hydrogen ion. Next, let's understand what is the pH electrode. What is the work of the pH electrode? The pH electrodes are constructed from a special composition glass which senses the hydrogen ion concentration. This glass is typically composed of alkali metal ions, and the alkali metal ions of the glass and the hydrogen ions in solution undergoes an ion exchange reaction which generates a potential difference. In a combination pH probe, the most widely used variety, there are actually two electrodes in one body. One portion is called the measuring pH electrode and the other is the reference electrode. The potential generated at the junction site of the measuring portion is due to the free hydrogen ion present in the solution. Now let's understand the standard operating procedure for measuring the pH meter, for doing work on the pH meter. First, what we have to do is we have to rinse the electrode with deionized water and blow dry using a piece of tissue paper. Next is to place the electrode in the solution of pH buffer. Allow the display to stabilize and then set the display to read 7 by adjusting the calibrating knob. Remove the electrode from the buffer. Again, you have to rinse and blow dry as before you put the electrode in the buffer of either 4 or 10 pH, whichever is near to the desired pH of the solution to be used for the experiment. Whatever you want to measure the pH of the solution, according to that you choose the buffer solution for measuring. Allow the display to stabilize and then set the display to read 4 or 10 by adjusting the calibrating knob. Adjustment of pH of experimental solution. Rinse and blow dry the electrode and put it in the experimental solution. Allow the display to stabilize and adjust the pH of solution by adding buffer component. Okay, these are the steps which you have to perform on a pH meter which has been already calibrated before. Okay, you in your lab you must have your pH meter which is always set to the which is properly set or accurate. So once you dip the electrode probe into the solution, it will automatically read into the if pH 7 solution, it will read the exact reading 7, and then for next buffer you can choose it. But there are some steps in which you don't have the accurate, sometimes you don't have the accurate reading of the pH. So for that I'll be discussing for the calibration of the pH in later on in this lecture. These are the steps for that pH meter which are already set, which gives the accurate reading. But if somehow the knobs are disturbed or somehow you won't get the accurate reading what you need, then I'll tell you how to calibrate it in later on. Okay, use of KCl. What is the use of KCl? Why KCl is used in the pH meter? KCl actually is a salt in nature which shows that it has a cation of a strong base and anion of a strong acid, and they both have no effect on the pH paper, and it has a standard value which shows that its pH is 7, which is a neutral figure. KCl is used as a salt bridge because it provides positive K-ions and negative calcium ions. The salt bridge is required to maintain the neutrality in the system but providing enough negative ions equals to the positive ions during oxidation. Now, how you can maintain the pH meter? Always keep the pH electrode moist. It is better to store electrode in a solution of 4 molar KCl. If 4 molar KCl is not available, then use a pH 4 to 7 buffer solution. Do not store your electrode in distilled or deionized water, as this will cause ions to leach out of the glass bulb and renders the electrode useless. Okay, KCl, as I have told that KCl is a neutral figure. That is, its pH is almost 7 or accurate 7. That is why the electrode is always kept dipped in this solution. What is the use of buffer solution, or what is a buffer solution? Buffers are solutions that have a constant pH values and the ability to resist change in that pH level. They are used to calibrate pH measurement system. They are can be small differences between the output of one electrode and another, as well as the changes in the output of electrodes over time. Therefore, the system must be periodically calibrated. Buffers are available with a wide range of pH values, and they come in both pre-mixed liquid form or as convenient by dry powder capsules. If you are getting the dry powder capsules, then what you have to do is you have to dissolve that capsule uh, in a deionized water or distilled water up to 100 ml, 100 ml. Dissolve it and make it a buffer solution of that particular pH. Most pH testers requires calibration at several specific pH values. One calibration is usually performed near the we can say isopotential point, the signal produced by an electrode at pH 7 at 25 degrees Celsius, and the second is typically performed at either pH 4 or pH 10. It is the best to select a buffer as close as possible to the actual pH value of the sample to be measured. Suppose if you want any if your solution is of some acidic in nature and you want to calibrate its pH, then you have to choose a pH 4 buffer. Otherwise, if your pH is somewhat basic in nature of that solution, your particular solution is of reading up to 6 pH, then you have to choose the pH 10 buffer. Okay, let's understand what is a pH calibration. Calibration is a measurement technology. It is the process of configuring an instrument to provide a result for a sample within an acceptable range. And metrology is the comparison of measurement values delivered by the device under test with those of a calibration standard of known accuracy. The pH meter measures the difference in electrical potential between the pH electrode and reference electrode, as I had told you earlier. For pH meter to work efficiently and to be accurate, they have to be properly calibrated. The meter is accurately translating voltage measurement into pH measurement, so they usually need testing in between or occasionally testing. Okay, let's see the pH calibration steps. Earlier I have told you the standard operating procedure in which your pH meter is already set. You don't have to set the values or you don't have to set the knobs for calibration. But if somewhat your calibration is disturbed or your knob is being disturbed and you won't you are not getting the actual reading for that particular buffer for 7 for 10, then you have to calibrate it again. For that, there are some steps that should be followed by you. Let's see. The first, you have to set the temperature. The temperature setting on the meter must be correspond to the temperature of the buffer used, or an automatic temperature compensator must be employed. Suppose generally what happens, all the buffers are usually kept in refrigerators. So whenever you want to calibrate a calibration, you have to do, if your buffer is of some cool temperature, then you have to set the temperature knob to 0 to 10 degrees Celsius or 10 to 20 degrees Celsius. And if your buffer is of as at room temperature, then you have to set your room temperature for about 25 degrees Celsius. You have to set the knob of the temperature. Now turn pH meter to pH or ATC if automatic temperature compensation is used. Next is to place clean electrode into fresh room temperature pH 7 buffer. Adjust the pH reading to exactly 7 using the 0 offset, standardized or set knob. Now rinse the once you have set the knob to the 7, you are getting the 7 reading in on the display. Then rinse the electrode with the distilled or deionized water. Blot dry it with the tissue paper. This would be the procedure for 1 point calibration. Continue through step 8 for the 2 point calibration. Now what happens? Once your knob is set to 7, you have to rinse your electrode with the distilled water, blot dry it with the tissue paper. Again, you have to place the electrode into the second buffer. Either you can choose it pH 4, or you can choose it to pH 10. Now adjust the pH reading to display the correct value using the slope, calibrate, or gain control course adjustment. What happens during in digital or pH meter, we have to adjust the slope. Adjust the pH reading to read the correct value using the slope knob. This is the fine adjustment. Once you are getting the reading of your pH around that somewhat, what happens if you have corrected your first dip into first reading as to 7, then in second calibration you have to set the knob to fourth. If you are taking the fourth buffer, you have to set the knob to fourth pH. Now at this time you have to set the knob the slope reading. If you are getting the slope adjustment to the accurate 4, minute difference could be manageable. Suppose if you are getting 4.05 or 4.10, up to 0.10, that should be okay for any of the pH meter. The calibration should not exceed. Suppose if you are maintaining the slope of the pH meter, then the slope reading should not be increased by 1.5 or by 4.20. The plus minus would be in between the 0.10 up to. Once your calibration steps have been accurately set, then what you have to do is you have to take your sample, which has to be calibrated, and okay, once you have set your pH calibration meter is set, then you have to take your sample to which you want to calculate the pH of that solution. You have to firstly rinse your electrode with the deionized water or distilled water, blot dry it, then again dip it into the particular solution. If you are getting the accurate reading for that particular pH of the solution, then it should be understood that the pH meter is properly set. Okay, now once you have calibrated your pH meter, let's understand what type of solutions are used for pH calibration. pH calibration solutions, also called the pH buffers, we have discussed it in earlier slide. These buffers are high grade buffers and are used to calibrate pH meter before each use is to get the most accurate result every time. These buffer solutions are used to calibrate pH meter because they resist changes in pH on addition of small amounts of acid or base. Suppose if you want to maintain any of the pH of the solution, particular solution, unknown solution, you have to make your pH for you have to make a solution for 5 pH, some acidic pH. Then you have to add some basic solutions into that, so that the particular pH comes across to nearby 5. That is why the buffer solutions are used to calibrate this. They are easily prepared for a given pH, and they are stable for long period of time. Next is the application of pH meter. There are various applications for pH meter. Nowadays, pH meters are generally employed in various industries, in pharmaceutical industries, cosmetic industries. We have to maintain the pH of every chemical that should be used in the drug manufacturing products. Okay, let's see. The rate and outcome of chemical reactions taking place in water often depends on the acidity of the water, and it is therefore useful to know the acidity of the water, typically measured by means of pH meter. Mostly all the industries requires the input of the water. None of the industries are not possible to run without the supply of water. Therefore, before using any kind of water, we have to check the pH. Sometimes what happen, the pH of the water differs from for different location. If we take the water of say location in Gujarat, the pH is somewhat comes around the basic in nature. It would maybe approx 8 to 9, whereas taking the pH of what whereas taking the calibration of the water in different areas, the pH would be differ for that different areas. That is why we required a pH meter for every industries, for any supply of water or for any use of water. Next is the knowledge of pH is useful or critical in many situations, including the chemical laboratory analysis. The pH meter are used for soil measurement in agriculture, the water quality for municipal water supplies, the swimming pools, the environmental remediation, the brewing of wine or beer, the manufacturing of healthcare and clinical applications such as the blood chemistry and many others applications. The advances in the instrumentation and in the detection have expanded the number of applications in which pH measurement can be conducted. The devices have been miniatured, enabling direct measurement of pH inside a living cells. In addition to measuring the pH of liquids, specially designed electrodes are available to measure the pH of semi-solid substances such as foods. These have a tips suitable for piercing semi-solid surface and have a electrode material compatible with the gradients in food and a resistance to clogging. The pH meter is also applicable for various research laboratory labs. Thank you for being with us. I hope you have enjoyed the lecture today. For more information, please log on to our website www.itlsacademy.com and register yourself for the summer training program.